Cool. Okay, so today we're going to be talking about, oh, this is not working, bacteria. Um, so today's lecture is on bacteria. I think this is a really good topic. I think the examiners like it quite a lot as well. In second year exams, at least from my experience, we had, I think, 3 out of 10 questions were on bacteria last year. So I'm not saying it would be the same again, but I do think it's a popular topic for examiners. So we're going to be talking about mainly bacterial structure, pathogenesis, and then treatment. So antibiotics and then antibiotic resistance, because I think that covers most of the syllabus points. I won't be talking about the tests for bacteria, but I think that those are quite easy to go through alone as well. So before we start, I'll just define some key terms. Um, just some simple things that could come up in the multiple choice questions that you should be able to answer quite quickly. So a commensal is just a harmless member of the flora, so that's like the gut microbiota. A pathogen is a microorganism that's able to cause disease. Um, Opportunistic versus primary infection. So basically, primary pathogens will cause disease regardless of the host environment, so internal environment, while opportunistic pathogens will only become pathogenic when there are certain conditions in the host. So, for example, the most common I think is like when the host is immunocompromised. Um, like with salmonella and HIV. HIV is a virus, but I'm just adding that in because a pathogen isn't necessarily a bacteria. It can be a virus or other things as well. So virulence is the degree of damage that a pathogen can inflict, and then the infective dose is the number of microbes needed to cause disease. So, for example, both Shigella and Salmonella can cause similar symptoms, but they have vastly different infective doses. So Shigella has an infective dose of less than 100, while Salmonella is, as you can see, more than 100,000. Basically, that means that Shigella requires less microbes to cause disease in a host than Salmonella does. Does anybody know what type of bacteria Shigella and Salmonella are? So gram positive or gram negative? You can unmute or put it in the chat, or I can or I can say it. Oh, so they're both gram negative. I think they're both gram negative bacteria. Um, so just a brief recap on bacterial structure. So this diagram I think it's just a really nice, concise way of showing the structure of a gram-positive bacterium and a gram-negative one. I think it's quite important to have like a simple diagram in mind for the exam as well, because um, it saves you a lot of words if you just draw it out, and it's easier for the examiners as well. So you can come back to this because these slides are uploaded, but this is basically the different structures of gram-positive and gram-negative. And then this table is going into a little bit more detail. So the difference between a gram-positive and gram-negative bacteria are basically, so the membrane in a gram-positive is single, whereas the gram-negative bacteria have double membranes, because gram-negative bacteria have an outer membrane as well, while gram-positive bacteria have a thick peptidoglycan cell wall. It just says that should say cell wall, and a gram-negative bacteria has a thinner one. Um, Teichoic acids, I think that's how you pronounce it, are only present in gram-positive bacteria and they're absent in gram-negative, whereas the opposite is that lipooligosaccharide saccharides are only present in gram-negative and not gram-positive. So those are just some simple differences that I think that you could mention quite quickly. If you recreate a table like this, then it would be quite easy for you to do, I think. In terms of gram negative, so now I'm just going to go over gram positive and gram negative bacteria separately in a little bit more detail. So as I said before, gram positive bacteria have a much thicker peptidoglycan cell wall layer. Um, there's also a difference between in gram staining. When you do gram staining, gram-positive bacteria stay purple because of the thicker layer, and they're able to retain the violet iodine complexes, whereas gram-negative bacteria aren't because their PG cell wall is thinner. basically, peptidoglycan is a polymer. It's made up of one 4-linked N-acetyl glucosamine and N-acetyl muramic acid. So those are two components that make it up. And another difference between gram-positive and gram-negative bacteria that I think I feel like it's a small difference, but people forget it quite often, is that the amino acids involved in the cross-linking between the peptidoglycan strands in the cell wall are different. So gram-positive bacteria uses L-lysine as the amino acid involved in the cross-linking, whereas gram-negative have mesodap. So gram-positive bacteria, their thicker peptidoglycan cell wall basically makes them a bigger target for beta-lactams than gram-negative bacteria. These are antibiotics that we'll discuss in a bit. And then these also lack an outer membrane, as I said before, and that's also important in terms of antibiotic susceptibility, which we'll talk about a bit later as well. So another thing, as I said, teichoic acids, I hope I pronounced that right, they're only present in gram-positive bacteria. So these are involved in cell wall metabolism. And I've just listed a few things that they're involved in. So adherence to the nasal epithelium. And there's two types of teichoic acids. There are wall TAs and lipo-TAs. So as their name suggests, wall TAs are linked to the cell wall layer, while lipo-TAs are linked to the lipid membrane through an extension. And basically, these can provide protection from antimicrobial peptides, which are produced by professional phagocytes. So basically, they provide protection because you can modify their charge. So, for example, I think I've given an example of D-alanine residues from DLT ABC gene products. These can make this, if when they attach to the TAs, they can make the structure more positively charged, and this can repel the antimicrobial peptides. And the charges of the wall TAs can also be modified to alter sensitivity to antibiotics this time, not phagocytes, because wall TAs are anionic and they can bind metal ions, basically cations, to increase cell rigidity and to basically to reduce the amount of antibiotic like molecules that can actually get through. So they're limiting the pores. Gram-negative bacteria now, so the difference, so as I said, they have an outer membrane, which gram-positive bacteria lack. This outer membrane is formed of two components, an inner layer and an outer layer. The inner layer is a phospholipid layer, whereas the outer layer is a glycoprotein layer, mainly lipooligosaccharide, as I mentioned before. So lipooligosaccharide is really important because it decreases membrane permeability. So it's basically it's another mechanism to sort of protect the bacteria against antibiotics because hydrophobic antibiotics won't be able to get through as much. And examples of these are like aminoglycosides or macrolides because they can't enter through the lipid membrane pathway because of these. Examples of, does anyone have any examples of aminoglycosides or macrolides? We'll go through them later anyway, so I'll tell you them later. This is also the glycoprotein outer layer is a target for insertion of the MAC complex and bacterial lysis by complement. So it is a target for the immune system essentially. It is something that can get the bacteria targeted as well. So an important thing that the outer membrane has are porins. So these are pore-forming proteins of specific size exclusion properties, and these allow the outer membrane of the bacteria to act as a selective barrier to like antibiotics, for example. So it's another way for the bacteria to decrease permeability to antibiotics. And this is really important because hydrophilic bacteria, as I said before, hydrophobic bacteria ability to enter is decreased by the outer layer, but here hydrophobic antibiotics, the ability of them to enter through the outer membrane is decreased because of lipooligosaccharide, but pores are important for hydrophilic antibiotics, and so they can enter through, and this is a way that they can wait, I've confused myself. Yeah, so pore expression is very important for hydrophilic antibiotic entry, essentially. Tetracyclines are an example that I've given of an antibiotic that relies on porin expression. And so basically the bacteria can mutate to decrease the ability of these to enter because, for example, Neisseria meningitidis, um, if you mutate one of the two major porins, you can make them resistant to tetracycline, and it's a big mechanism of antibiotic resistance in these bacteria. And also the outer membrane can form outer membrane vesicles, which help with endotoxin release and have immunomodulatory effects. So another thing that I mentioned earlier was that gram-negative bacteria have a much thinner peptidoglycan cell wall layer, so they're much less affected by beta-lactams. So Mesodap, as I said, is involved in the cross-links between the PG strands, and NOD1 is a pattern recognition receptor. That's what PRR stands for, which detects Mesodap from gram-negative bacteria like Neisseria meningitidis, which increases inflammatory cytokine release, which is a characteristic of Neisseria meningitidis. So this is why this inflammatory cytokine release happens. And the thinner wall means that they're more prone to sort of spreading by desiccation, essentially. So now I'm going to move on to bacterial pathogenesis. Um, so the key traits of a pathogen. Um, so these five sort of topics are five subsections are important to bacterial pathogenesis. And if you were writing an essay, I think that it would be good to structure it in this way. So to talk about these five subsections because they're all important to pathogenesis. So tropism, replication, evasion of the immune response, damage, and then further transmission. Like a bacteria needs to be able to do all of those to survive and be a successful pathogen. So I think we'll talk about tropism. Oh, and then you also have Koch's postulates. I'm not sure if I said that name right. So these are also very important. Basically, I think this is on the syllabus, I'm pretty sure. Basically, this is talking about how you identify a pathogenic microorganism. So the microorganism needs to be found in all organisms suffering from disease. You need to be able to isolate it. It shouldn't be found in healthy individuals. And once you isolate it, you need to be able to grow it in pure culture. And then if you reinfect someone or a healthy person, maybe not a person, a healthy organism, then that should cause disease in that organism. And um, and then when you isolate the disease from the organism that's become deceased because you introduce something to it, that should match the microorganism that you previously isolated earlier. That's just basically it's criteria for how you know a pathogen is involved in an infection. And then there's also some molecular postulates, which is essentially the same thing, but dealing at a molecular level. So if you isolate I think if you isolate the gene from the pathogenic strain, if you knock out um the 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 gene involved, then it leads to the loss of the phenotype. But if you then knock in that gene into genetic element into a strain which is not infected, it should become pathogenic essentially. But these are nice diagrams, which I think are helpful. So back to pathogenesis. Tropism is pretty standard. Basically, it's talking about finding and establishing a niche, so an area that the bacteria can infect in the host that would be most beneficial to them, which would be basically where they have a higher chance of becoming successful. So some bacteria are intracellular and some are extracellular. The advantage of having intracellular bacteria is that they can be shielded from the host's immune response a lot more than extracellular bacteria. So for example, white blood cells or like antibodies and stuff, it's easier for them to survive that. So being an intracellular bacteria is also an immune evasion technique as well, if you want to mention that. So the two examples I chose were Shigella and Salmonella. These these enter the cell using T3SS, which is basically like this needle-like thing, which helps them enter the cell. And they survive in a phagolysosome, which I'll talk about more in just a few seconds. And basically, this being intracellular, when they're intracellular, they prevent activation of the inflammasome as well, so that they don't trigger the immune system. And extracellular bacteria example are streptococci and staphylococci. E. coli, as you can see, there's a bunch of examples on there. They have to attach to a surface, and that's how I think by adhering to a surface, they try and prevent dying by the immune system because they need to be able to adhere to something so that they're not swept away. It's a really bad way of saying it. And then replication, there's not much to say really. They just need to be able to replicate or obtain nutrients. They need to be able to feed themselves and form more of themselves to be successful because as you saw earlier, the infective dose is different for each bacteria, but they need to reach that infective dose to be able to be a successful pathogen. So now just a really quick thing about transmission. So there's three main different types of transitions: horizontal, vertical, and zoonotic. So the last one is about through like mosquitoes and vectors. Horizontal transmission can be human to human or non-human to human. So like, like respiratory, for example, if you're infected by something and you cough and it's like an airdrop infection, that's an example. Fecal oral or sexual contact. Non-human to human, as in like animals to human. Vertical transmission is from mother to child. So that can happen when the mother is feeding the baby, breastfeeding the baby through the milk or through the placenta, actually, when the baby is in the womb, and the fetus is in the womb. And then I've already talked about the last one. So adherence, as I was saying, it's important to be able to adhere to the environment that they've chosen for bacteria because they need to be able to resist removal and subsequent destruction. Molecules that adhere to the surface are called adhesins. So there are a few different mechanisms of adherence. So some bacteria produce pili. A good example is uropathogenic E. coli, as I've said here, because it has two different types, type 1 and PAP. So it depends on where they are. They use different types of pili, basically. So if they're in the urinary epithelium, they can use type 1. And if they're in the kidney, they use P, the P type 1. So on these, they have a FIMH tip, which binds to mannose-related uroplakins on the bladder epithelium, and that basically helps it adhere to the bladder epithelium and resist removal, essentially. And so FIMH inhibitors are a way to prevent E. coli infection, so a potential way to prevent it. And then different other adhesins are things like capsules or the glycocalyx. And then I've included an example there. But basically, it's really important, especially for extracellular bacteria, to be able to adhere to their environment because otherwise they'll be removed by the immune system and they won't be successful in their pathogenesis. Other examples are biofilms. Biofilms are really important. I think I am pretty sure they're an important part of the syllabus as well. And they're really interesting to talk about. And there are a lot of pieces of experimental evidence that you can find on them. So I think this is a good thing to expand on in the adherence section. They're basically self-formed extracellular matrices forming communities of bacteria. So they can form on foreign bodies as well. So this is really beneficial because phagocytes can't attach because of different binding proteins and stuff. They can form on like prosthetic joints, heart valves, intravenous catheters, and this really helps them evade the immune system basically because phagocytes can't go bind to those either. They can also be found on native structures like the heart. So basically, they protect bacteria from antibiotics, immune defenses, as we've talked before, and yeah, phagocytes are poorly able to adhere to it, contributing to immune evasion. I'm pretty sure they also help with antibiotic resistance because it's just forming a community of bacteria, so it makes it much easier to spread antibiotic resistance between them. Yeah, so they they prevent good wound healing, which can cause chronic infections. And then this is a really good piece of experimental evidence, the one that I've got down there on Staph aureus, Ryder, Chopra, and O'Neill. Um, I think yeah, it's really nice to explain and it's really well done as well. So they grew Staph aureus planktonically and with biofilm, and they found that there were more mutants, so more mutants that caused antibiotic resistance when the biofilm was present than without. And when they cultured it with an antioxidant, this reduced the mutation frequency. And that basically it's suggesting that the mechanism of action for more mutations happening with a biofilm may be related to greater oxidative stress because the antioxidant reduced mutation frequency. But that's a really good one if you want to look it up. And then biofilm production is controlled by quorum sensing. So it's basically that bacteria can sense when the number of bacteria and the environment is optimal so that they can spread more. So the biofilm can be produced at a time when it's most beneficial to the bacteria for causing infection. And then intracellular bacteria and extracellular bacteria have different, it's invasion. I think it should say immune evasion. Sorry, I think this should say immune evasion. But they have different, wait. No, it is invasion. Sorry. Yeah, so they have different mechanisms because obviously they're invading different things. So for intracellular bacteria, they obviously have to get inside cells. And then, as I said before, this enhances immune evasion because they're away from antibodies, neutrophils, the immune system, essentially, a lot of the immune system. So they can be taken up by a phagosome once they enter, and they have to prevent a phagolysosome forming successfully. So because if that forms, then they'll be destroyed by the immune system. So they can do this in 3 different ways. And I put a diagram in here which shows this really well. You could just put that in the exam in an essay. So the first way is like by inhibiting fusion of the phagolysosome with the phagosome with the lysosome, and that avoids the degrading enzymes in the lysosome from degrading the bacteria. And basically preventing lysosome function. And Mycobacterium and Legionella do this. Another way is that you could inhibit acidification of the phagosome, which would also decrease activity of the enzymes. But the main function here is to stop the lysosomal enzymes from destroying the bacteria. And then the third one is that the bacteria could escape from the phagosome into the cytoplasm where there are no enzymes present. And those are the 3 ways that they can basically evade immune defenses. So for extracellular bacteria, instead of getting inside a cell, they may need to breach like the basement membrane of tissues. They do this by having like enzymes like collagenase, which can and these can degrade important aspects of the basement membrane to allow spread through the tissue, and this is important in like cellulitis caused by strep pyogenes. Staph aureus produces a hyaluronidase, which invades and digests the basement membrane. It also produces exfoliatin, which disrupts desmosomes, which, if you remember from first year, are really important for the skin, in the skin layers, keeping skin together. And basically, this can lead to scalded skin syndrome, like a lot of rashes, wrinkly skin sort of falling off because skin starts shedding because you've disrupted the desmosomes. And then ICAM1 is on endothelial cells. Extracellular adhesion proteins from the bacteria can bind to that and prevent leukocytes getting in extravasation. Basically, it makes it harder for the immune cells to be recruited to the site of action, preventing immune response, essentially. So everything that bacteria are doing is to A, be successful in getting to where they need to get to, and B, evade the immune system so that they can survive long enough to cause infection. And then I've just got a few more examples of things, enzymes, which could be used, like IgA protease degrades IgA to allow adherence to mucous membranes, like in the nose, for example, or other places. So these are mainly produced by Neisseria gonorrhea, Haemophilus influenza, and Streptococcus pneumoniae. And then you also have leukocidins, which can destroy neutrophilic leukocytes and macrophages. So basically, this is all on slides, so you can look through it later. And now finally, we've got onto immune evasion. So immune evasion, there's a range of different things that bacteria do, and to be successful, pathogens. So you can have capsules, which are basically high molecular weight polymers. They exist external to the peptidoglycan cell wall layer, and that they prevent the phagocytes from adhering to the bacteria. And they also block underlying access to like PAMPs, which could trigger, pattern recognition receptors, leading to phagocytes arriving and an immune system response. And this is basically the first line of Staph aureus defense. Capsules also prevent complement binding, so they prevent complement binding, opsonization, and MAC formation. And then they're also important in molecular mimicry. For example, Neisseria meningitidis, the serogroup B, is structurally similar to NCAM1, which makes it poorly immunogenic. And that's a reason why there's no vaccine available / produced against the capsule yet, because serogroup B is a big part of the capsule. Whereas there is one against serogroup C. Protein binding factors are another way to evade the immune system. Neisseria meningitidis is factor H binding protein, which binds to factor H, preventing alternate complement activation. If you remember, factor H is quite important for that. And then you also have the biofilm, as I mentioned before. That's important for antibiotic resistance because the community of bacteria that forms can share virulent factors. Basically, resistance can spread a lot easier. And then another way to evade complement is protein A of Staph aureus binds to the Fc region of IgGs and antibody, preventing activation of complement. And you also have RNA thermosensors, which I think are quite exciting. And I've included a piece of experimental evidence by Tang et al. 2013. These are found on the 5' UTR region of mRNA, and they allow thermoregulated immune evasion. So essentially what that means is when higher temperatures occur during fever because of infection, the bacteria is more likely to survive. And it can like elicit immune evasion factors when that happens and the host is already immunocompromised and stuff. So it's helping the bacteria to be more successful. So toxins are very important. So toxins are basically how they drive toxins are factors which drive pathogenesis and infection. They can either cause the infections, the symptoms directly themselves or through a host response. There's two types, endotoxins and exotoxins. Exotoxins are polypeptides released by cells. These are formed by both gram-positive and gram-negative bacteria, and then endotoxins are gram-negative bacteria. Yeah, so we'll talk a little bit more about them. Yeah, so exotoxins, as I said before, they're secreted by both gram-positive and gram-negative bacteria. They are polypeptides, and they're good antigens. They have good antigenic ability. A lot of them are used in the production of antitoxins and vaccines. So basically, you can treat them with either acid, heat, or formaldehyde, and you can convert them to toxoids, which can then be used in vaccines because they're no longer toxic, but they still retain the ability to induce protective antibodies. And these are successful vaccines as well. So the tetanus vaccine, which requires a booster every 10 years, and the diphtheria vaccine, which doesn't require a booster if the dosage schedule has been followed in childhood. There's a few different types of exotoxins, and I'll just briefly go over those. AB toxins are one example. So basically in AB toxins, the A subunit is toxic, while the B subunit mediates binding to the specific receptors. So like tropism, basically, it leads to the the exotoxin to where it needs to be, and then the A subunit is the toxic component, which can drive pathogenesis. Enterotoxins are an example, Vibrio cholerae. The B subunit binds to GM1 ganglioside on the cell memory of enterocytes in the small intestine. And then the A can enter the cell, cause ADP ribosylation of the GS factor, leading to an increase in adenylyl cyclase, CAMP, and leading to the opening of ion channels, which causes an extra secretion of um, various ions. Basically, it causes excess secretion of ions and inhibits sodium absorption, um, and causes fluid and electrolyte loss in the gut lumen. It causes diarrhea basically. And then you also have heat labile enterotoxin by E. coli. As another example. These are just examples that you could expand more on after you look them up in a bit more detail, I think. And there's a few pieces of experimental evidence that you could also find on these, from what I remember. Pore-forming toxins are another type. These bind to receptors. They bind to receptors, increase in concentration, and then oligomerize to form an influx pore, influx or efflux pore, killing cells. So basically, it causes a loss of ions or electrolytes essentially through the pore, and this can lead to the cells dying. And an example is there as well. Now, another type are proteases. I think these are the last two, proteases and superantigens. Protease activity. So what I mean by that is some bacteria have proteases involved, which can break down proteins and cause the infection. So exfoliating, as I talked about before, it cleaves desmoglein in desmosomes, leading to scalded skin syndrome. And it causes rashes, skin peeling off, and tissue paper like wrinkling of the skin. And it also allows Staph aureus to spread through gaps in skin cells because, as I said, desmosomes help hold the cells together. Neurotoxins are another example of this because, like botulinum toxin from C. botulinum, this blocks as acetylcholine release at the neuromuscular junction by cleaving the snare proteins, which, if you remember from first year, are important for vesicle release of ACH. And then you also have the tetanus toxin, which blocks GABA and glycine release to prevent muscular relaxation. So the tetanus toxin basically travels, I think, in a retrograde fashion back to the brain. Yes, may need to look that up. So superantigens are the last type. They cause an excess and non-specific activation of T cells, leading to massive cytokine release. For example, Staph aureus, you have TSST1. This attaches to the beta domain of the TCR, which is the T cell receptor, linking it to MHC2 of the antigen presenting cells, which causes toxic shock syndrome, essentially, and leads to a massive inflammatory response, ultimately ending in multi-organ failure. So superantigens are pretty big, pretty bad. And now on to endotoxins. So endotoxins are liposaccharides. They're poorly antigenic, so you don't have any you can't form vaccines against them because they induce protective antibodies really poorly. And multiple episodes of toxicity can occur. They have a lower toxicity compared to exotoxins, but they also can't be used in vaccines. So there are different modes of actions for endotoxins, and you can actually make a really nice diagram with this. I remember that I made a really nice diagram in the exam for this. So they can do three different things ultimately. Activate macrophages by binding to CD14, activating TLR4. This leads to the release of cytokines such as IL-2, TNF, and then also the release of nitric oxide. So IL-2 can act on the hypothalamus to induce fever. TNF can increase capillary permeability, leading to hypotension. And then nitric oxide causes vasodilation, as you know, leading to hypotension as well. They can also I think work through the alternative complement pathway. So C5a um, can induce neutrophil chemotaxis, leading to inflammation and tissue damage, and C3a lead to hypertension and edema. And then tissue factor is also involved. So basically endotoxins can lead to systemic activation of the coagulation cascade, um, essentially leading in multi-organ failure. Basically, all these things characterize a systemic inflammatory response syndrome, and there are five main features of that: fever, hypertension, tachycardia, tachypnea, and leukocytosis. Um, yes, so they're all caused by endotoxins, like the endotoxin specifically. So if you purify LPS, if purified LPS is taken out of bacteria, it can reproduce these effects without the bacteria needed. So this can be blocked by antiserum against endotoxin, even though you can't make vaccines. So three components comprise an endotoxin. You have lipid A, O antigen, and a core polysaccharide. Lipid A is a toxic component, and then Westphal and Luderitz, that is a good piece of experimental evidence if you look that up. They demonstrated that lipid A was a toxic component quite well. And then you have the O antigen, which is a polysaccharide on the exterior, and it also has repeated sugar, made up of repeated sugar. And this is different. This is not conserved. It differs between different species, and frequency differs between different strains of the same species of bacteria, whereas the core polysaccharide is conserved in bacteria. So endotoxins, the main, like, the big bad thing that they can lead to is septic shock. As mentioned like 2 slides ago, they can lead to fever, hypertension, tachycardia, tachypnea, leukocytosis. Um, Essentially, these are all things which you can see in septic shock as well. And septic shock is a leading cause of death in the ICU. Um, for septic shock and toxic shock are different because bacteria are in the bloodstream, whereas the toxin is circulating in toxic shock. So toxic shock is another type of shock. Septic shock has positive blood cultures because the bacteria are in the bloodstream, whereas toxic shock has negative blood cultures. So that's a way to tell the difference between the different types of shock if a patient is suffering from it. Um, and then septic shock can kill the patient even if antibiotics have killed the bacteria because it's mediated by cytokines like TNF and interleukin 1, which can continue even after the bacteria have been killed. So it's quite dangerous in that way. And Neisseria meningitidis is a really good example of septic shock. So it can lead to acute fulminant meningococcal septicemia. And this happens mainly in young children, and it causes a bunch of things: cardiovascular collapse and then death within 12 hours. So it's really dangerous. And then this is just a little table of toxins, good examples of toxins that you can probably use and look up to go more in detail with. One of the questions that came up in our exams last year was just on bacterial toxins, so they can just have an entire essay question based on bacterial toxins. So it's important that you have examples ready to be able to talk through the different types, like all the types that I've gone to. So endotoxins don't have different types per se, whereas exotoxins have all of these, like the AB toxins, pore foaming, superantigens, proteases. So you need to have an example for each of those, I think, and then a couple of examples for endotoxins. So now we'll just move on to the final part, which are antibiotics. So you can treat bacterial infection using antibiotics. Um, you have a range of other things you can do. So physical interventions, as well. But antibiotics are the main treatment. So they have a number of different mechanisms. They can be cell wall inhibitors, which target the peptidoglycan cell layer. We were talking about beta-lactams earlier. They can be protein inhibitors because the ribosomes present in bacteria and humans are different. Bacteria have 30S and 50S ribosomes, whereas humans have 40S and 60S, which allows efficient targeting because antibiotics do need to be selective to the bacteria, or you don't want human cells to be targeted as well. And then they can also be DNA synthesis inhibitors. So they can prevent nucleic acid synthesis. For example, some more examples will be mentioned later on, and they can also be inhibitors of folic acid metabolism because the process is different between humans and bacteria. Antibiotics can either be bactericidal or bacteriostatic. So bactericidal basically means they kill the bacteria, but bacteriostatic means they prevent the growth of bacteria. So if you stop taking stop taking the antibiotic and it's bacteriostatic, then the bacteria can start growing again and cause infection. So broad or narrow spectrum antibiotics exist. Normally in treatment, you start with broad and then narrow it down to narrow spectrum because when you start off, you may not know the specific bacteria that's causing the infection. So, and broad spectrum antibiotics can work against many bacterial types. So they're very important for initial treatment when we're unsure what bacteria is infecting the patient. But then you want to narrow it down so that you can, once you've identified the bacteria through cell culture, you can narrow it down using narrow spectrum, which is better because it prevents antibiotic resistance, basically. So now I'll just go through a little bit on cell wall inhibitors, protein inhibitors, and then DNA synthesis inhibitors. Cell wall inhibitors, as I said, peptidoglycan is a huge component of bacterial cell walls, and it's only found in bacterial cell walls. So it offers selective toxicity when treating us like humans. They differ between so the susceptibility differs depending on whether the bacteria is gram-positive or gram-negative. As you can remember, gram-negative bacteria have outer membranes, so that sort of helps them against beta-lactams. The penicillins are, I think, one of the most popular examples of beta-lactam of antibiotics. Obviously, penicillin was found by Fleming in 1928. I'm sure most people know about that, taught in GCSE. But they basically block cell wall synthesis. They're bactericidal, so they do kill the bacteria. They can attach to penicillin binding proteins because they have similar stereochemistry to D-alanine and D-alanine. Alanine. And they bind to the beta-lactam ring. Yeah, so these are beta-lactam inhibitors. So they bind to the beta-lactam ring, preventing peptidoglycan cross-linking, which I mentioned earlier is important, and cell wall synthesis. So without that, the bacteria will die because it's an essential component of the cell wall and bacteria. Gram-negative are less susceptible because of the outer membrane, as I mentioned. These can be inactivated by beta-lactamases from bacteria, which is a method of resistance that bacteria have developed. But we can also work through this because you can make the side chain and modify the side chain to make it inaccessible to beta-lactamases. And you can also inhibit beta-lactamase using by using things like clavulanic acid. You can also use other adjuvants while treating with the antibiotic to inhibit the enzyme as well. Another example are glycopeptides. These block transpeptidation, which is an important factor of the peptidoglycan cell wall layer. They bind directly to the D-alanine bit, which inhibits the binding of transpeptidase. So they don't have similar stereochemistry to this. They bind directly to it. So basically preventing so basically preventing it from binding to penicillin binding proteins as well. So these are gram-positive specific, and they're used to treat MRSA. So for example, you can use intravenous vancomycin to treat it. And then I think this is the last example of cell wall inhibitors is isoniazid, however you pronounce that. This inhibits the synthesis of mycolic acids, which are important constituents of the mycobacterial cell wall. So mycobacterium are a type of bacteria. And they're also used in the treatment of TB alongside I can't say that, but you can see it on the slide. It's not a good topic when you can't pronounce half the things. So then you have protein inhibitors. These can bind to ribosomes and inhibit translation, which is really important in protein synthesis. If you remember from first year, I think. They are bacteriostatic though, so if you stop taking them, the bacteria will regrow, can start growing again. Selective toxicity, as I said before, because the types of ribosomes involved are different between bacteria and humans. Aminoglycosides are an example. They block the attachment of tRNA to the A site of the ribosome. So tetracyclines can do that. And then streptomycin works by preventing tRNA binding to the ribosome as well. And then gentamicin blocks tRNA and mRNA interactions. Essentially, they're just preventing translation, as I said before. Macrolides can inhibit translocation, and amphenolyticals can inhibit peptidyl transferase. So I've just put, those are just examples, by the way, that you could include if you're talking about antibiotics. It's a mix of the left-hand side and the right-hand side part of the syllabus. DNA synthesis inhibitors include folate inhibitors, so basically folic acid, as I said before. So essentially, bacteria and humans synthesize folic acid differently. I think humans don't synthesize it, they get it from the diet, whereas bacteria do, which helps with selective targeting. So I've just included a few examples on there of how they do this, essentially. Um, yeah, so you also have transcription inhibitors. These bind to DNA-dependent RNA polymerase. And then you have DNA gyrase inhibitors. So these can prevent like coiling of DNA, leading to a relaxed state, I think. So basically it's preventing DNA synthesis, essentially. And then you have to think of a few considerations for antibiotic use. Can it reach the infected site? Would it be better to use a physical intervention? What are they going to do on the body? Will they lead to hypersensitivity reactions? What side effects do they have? Do they affect the normal bacteria of the gut that are important? Is there any resistance? So I'll just talk about resistance now, which is quite important, I think. Horizontal gene transfer, it's just I've highlighted that because there are a few seminal studies which are really good to include. So Griffiths, 1928, is quite important as well. Basically demonstrated horizontal gene transfer. And if you see this diagram here, this is a really good diagram showing the different types of horizontal gene transfer. So you have conjugation, transduction, and then transformation. Conjugation is basically plasmid mediated. Transduction is like the release of like a phage which carries the bacterial genes, resistance genes, and this can infect the recipient cell, leading to a resistance. And then transformation is basically when there's bacterial cell death, when cell death or lysis, the free DNA is just released, and that can also be transported and taken up by other cells, leading to resistance. And then this slide just summarizes antibiotic resistance. So you have different types that you can have basically. I think in an essay, it's important to try and mention most. I think I would structure the essay as by sort of mentioning the resistance types while talking about the antibiotics, just to ensure that you've included them all. But it's still okay if you want to talk about the antibiotics first and then antibiotic resistance later on. So I think this is quite a nice table that I found. And then targeting antibiotic resistance. So obviously antibiotic resistance is a really, really big issue at the moment, especially because we don't have the production. We're not really producing more antibiotics and we're increasing resistance. So it's going to become a lot harder to treat bacteria, and you get multi-resistant bacteria as well. So we need to improve prescribing of antibiotics. Physicians need to be able to be given guidelines, I think, to decrease unnecessary prescriptions. These are just a few examples that you could list in an essay if it came up, if they ask how you could target antibiotic resistance. Stopping the over-the-counter trade of antibiotics. So in a lot of countries, antibiotics are just available for general sales. You can just go to the pharmacist and buy them without any prescription, which increases resistance because the likelihood of resistance, because it's more likely to be misused. For example, the course may not be finished because it's really important to finish the course of antibiotics, or they may be overused, which can also lead to resistance. Also, increasing regulation for antibiotic use in farming. Um, so I think it's a really large amount of antibiotics are used in farming as well to increase yield of animals and stuff. And the thing is, resistance spreads between animals, and that resistance can spread to humans. Like there's no such difference in antibiotic resistance in different species, if that makes sense. So one could be re increasing regulation or reducing animal farming, perhaps. Um, Discovery of new antibiotics would be ideal, but there's not a lot of incentive for drug companies, big pharma companies, because there's a lack of profit. They want to deal with chronic diseases because if you create a medicine for a chronic disease, you're going to be paid for a much longer time because it's going to be affecting the individual for a much longer time than a short-term infection. So that's one of the ways why incentive is reduced, but also because if new antibiotics are found, they'd have to be reserved because there's so many multi-drug resistant strains now. So the companies wouldn't be able to make a profit if they did find a successful antibiotic because those drugs would need to be safeguarded essentially. And also the drugs can become ineffective quite quick because of the rate of antibiotic resistance. So perhaps government schemes may help. And then there's a few, I've just included an example of a new antibiotic that's been discovered recently, which can be used in the treatment of multi-drug resistant and gonorrhea. Uh, so I think I talked about this before. These are resistant to quite a few antibiotics, and I think it's been found that if bacteria are exposed to this drug, they it can be effective against them. But again, like there's a lot of research left to happen. Then there's a few more examples online as well. So I think it's always important to use at least one example of a new one that's been discovered. Okay, so that is it for me. Does anyone have any questions?